Welcome to this edition of the Microbial School Bus, and today we're going to be celebrating the thrifting skills of bacteria and discussing how this benefits us. So do you think you're good at finding a bargain? Do you consider yourself gutsy because you hassle with sellers to get a better deal? Well, bacteria are even more bold. They will snatch the DNA from their bacterial colleagues as they lay dying and oozing out their cell contents. Yes, bacteria are ruthless, but this can be good for us. First, a little about bacteria. They're single cell and can come in different shapes, including round, which is coccus, rod or bacillus shaped, and comma or vibrio, just to name a few. They have one genome chromosome that's circular. So genome chromosome means they have to have that chromosome to survive. Their DNA is made up of the same chemicals as ours, just in a different order. So similar to how our alphabet can spell multiple different words, it's the same chemicals that make up their DNA, but those nitrogen bases, if you remember the A, T, C, G from biology, they're just in a different order. Bacterial cells are much smaller and less complex than ours. So here's our little stuffed animal E. coli, and I'll also put a picture up of a bacteria. But they have some abilities that our cells don't have. One of them is they can easily pick up DNA from a peer and start using it. So how does this happen? Well, let's say you use some cleaning spray in your home that kills 99.9% of bacteria on your counter or in your bathtub or wherever. Well, that means that a few survived, right? After all, there were probably millions of bacteria on your counter or in your tub before. Well, those few surviving bacteria are probably moving through the carnage of dead bacteria, and they might notice that some of those bacteria are leaking out their contents, and they might see DNA from those dead bacteria around there. Well, they don't need it anymore, right? Why not take it? And they do. They snatch that DNA from their dead friend and they start using it. That's brutal, and in this case, it might not be good for us. Sometimes extra pieces of DNA that bacteria can acquire, called plasmids, might contain genes that make the bacteria resistant to antibiotics and other chemicals, so using cleaners and antibiotics that you don't need isn't necessarily a good thing. It leaves only the strongest bacteria with no competition and allows them to enhance their repertoire of skills by picking up DNA or plasmids from their dead colleagues. How have we exploited bacteria's ability to pick up and use DNA that's not theirs? By making them produce medicines like insulin and factor VIII, a clotting factor needed by hemophiliacs, just to name a few. So how do we do this? We take a gene, which is just a small segment of DNA that codes for something we need, like insulin, and we take a special enzyme to attach that gene to an extra piece of DNA that bacteria often carry, like the plasmid I mentioned. And the process of attaching those two, that human insulin gene to the plasmid, is called recombinant DNA technology. And then we let bacteria do what they do, so we might get a bacteria like E. coli, and they will pick it up. Now, we can encourage bacteria to do this a little bit faster by either giving them a jolt of electricity or treating them with a chemical that will put a temporary hole in that bacterial cell wall so that they can pick up that plasmid faster. But this is a process that they already do. Once they have that plasmid in them, the bacteria will start expressing or using the plasmid with the insulin gene, and voila, we have insulin made by bacteria. The bacteria won't complain about it because you fed them and you kept them warm while they made the insulin, and you won't have PETA protesting your facility because you didn't hurt any animals. It's also cheaper and safer to have bacteria produce insulin because they don't take up a lot of space or food, and they won't bite or scratch you. All you have to do now is collect your insulin, purify it, and it's ready to be packed and shipped. Sometimes we use yeast to make insulin too, as in the same type of yeast used to make beer. On a side note, we've been using bacteria to produce human proteins like insulin for at least 40 years. Science is great, isn't it?